Hi Gen Chem Honors, we're Annie and Dominic, and our topic, the role of telomerase in cell immortalization and cancer treatment, aims to study
the structure and function of telomerase and its possible role in cancer treatment. As you see on your screen, this enzyme here is what we're going to be studying. And as you'll also see on your screen again, you'll see that this molecule here, 2,6-diaminoanthracnone, will be the molecule that's in charge of denaturing the G quadruplex inside telomerase, which is in the active site. So hopefully using those two components, we'll be able to develop a treatment for cancer.
So why do we care? Well, telomerase is the enzyme associated with cellular immortalization, and cell immortalization is associated with cancer. So those two things are inextricably tied, and if we can denature telomerase, we can potentially treat cancer. So as you can see in your figure here, a cancer cell is injected with a telomerase inhibitor, and as this is done, the cell undergoes apoptosis. Now, this is a positive implication of using the telomerase inhibitor, like a compound such as 2,6-diaminoanthracnone, because it targets the cancer cell by denaturing telomerase and it kills it, and that's exactly what we want to do. Now, on the left side of your figure, you'll see a healthy stem cell that's being injected with the telomerase inhibitor. It shortens its long telomeres, and once the anti-telomerase therapy is stopped, it becomes a healthy cell once more. So the implication here is that if you inject a healthy cell with the telomerase inhibitor, the telomerase also starts to denature. So it's not exclusive. This therapy is not exclusive to cancer cells that have telomerase. It applies to all cells that have telomerase. So there are both negative and positive implications of using this treatment, because we can either target and kill cancer cells, or we can target healthy cells unintentionally and denature their telomerase and have them undergo apoptosis as well.
Okay, so before we get into the telomerase itself, there's some background things that we need to cover. So the first thing that we want to discuss is DNA replication, more specifically what the leading strand and the lagging strand does. To explain this, I'm going to proceed to this figure right here. Just to note, the red strands are the original strands and the green strands are the newly replicated strands. Now, with the leading strand, it can be replicated in one continuous segment, so no DNA is lost in the replication, which is fine. But the key point of this conversation is what happens in the lagging strand. Going back to the figure, as you can see, in the second set of strands, you see these boxed in R's in these green segments. That's because the lagging strand can only be replicated in certain segments marked by RNA primers, as denoted by those boxes. And the problem with this is the very end RNA primer, when that's removed, that segment of DNA is not replicated. So if that's actual coding DNA, that could be potentially fatal to the cell. So this is where telomeres come in. So telomeres are basically repeated segments that are rich in guanine that are capped at the end of the lagging strand to prevent actual DNA coding degradation. So as you can see, there's a strong correlation between length and aging. So since telomeres protect the ends of the lagging strand during replication, the DNA that gets cut off is actually non-coding. It's just telomere, which gives the cell a certain number of life divisions it can undergo before actually reaching a critical length. So this actually leads into some fundamentals about the cell cycle we need to cover. So the first thing we need to talk about is senescence, or the M1 arrest phase, or mortality 1 arrest phase. For this, we proceed to another figure. As you can see, the telomeres, since they're only a finite length, after a certain number of divisions, called the Hayflick limit, which is the maximum number of divisions a cell can undergo before reaching senescence, the telomeres reach a critically short length, as you can see at the bottom of your screen. So once this happens, most cells stop dividing or enter the G0 phase of the cell cycle. But some cells can actually make it past this phase and enter what is called crisis, or the mortality 2 phase. And this is when cells' telomeres actually get even shorter and cells start to die in large numbers. But as we'll see shortly, some cancerous cells with specific properties can actually make it past crisis, which could cause a problem.
So in terms of the structure of telomerase, it is a RNA-dependent DNA polymerase, which essentially means that it uses RNA complementary base pairing as opposed to DNA complementary base pairing. So its catalytic core consists of two main subunits, HTR and HTERT, HTR stands for human telomerase RNA, and HTERT stands for human telomerase reverse transcriptase. Now, HTR provides the RNA template for HTERT to begin adding base pairs to the telomere sequence. And the main thing to take away from this figure, as you see, is that telomerase has a quaternary structure, which means that it consists of several smaller subunits, each of which is connected by certain intermolecular forces, such as hydrogen bonds, dipole-dipole attractions, dispersion forces, and within those subunits, there are disulfide bridges, which hold the actual subunits together. But the intermolecular forces are going to be important later on when we discuss the interactions between the telomerase and the 2,6-diaminone-thraquinone.
Okay, so as we already kind of mentioned, there's a few basic functions of telomerase. The first is catalyzing the telomere synthesis reaction, which basically means that it builds telomeres. This reaction involves four things. It involves primer recognition, base pairing, nucleotide addition, and translocation. So to explain these four things, I'm going to proceed to a figure, as you can see now on your screen. The first part of this figure shows primer recognition. So basically, the RNA sequence finds the complementary DNA sequence at the end of the telomere, and that tells it to initiate the reaction. Then from there, you see actual base pairing. Base pairing and nucleotide addition actually occur in the last three steps of this figure. And basically what that means is the RNA bases in the HTR template provide the base sequence that in the RNA code that complements the DNA sequence. So basically allows it to select which bases actually need to be synthesized. And then from there, nucleotide addition just basically means that all these green bases, DNA bases you see here, the new bases are bound together into a new strand telomere. And then as you can see, in the third step here, there's translocation. So once it finishes replicating, the enzyme jumps to the end and then restarts the process. So although we don't know how many times this actually occurs in a cell with telomerase activation, we do know that there are certain genetic mechanisms and catabolic mechanisms that regulate how much this reaction occurs. So knowing this, we can now conclude that telomerase plays a role in cell immortalization because since telomeres determine the lifespan of normal cells, if these telomeres were rebuilt every time they're shaved off during replication, the cell can essentially divide indefinitely.
So telomerase is not only found in cancer cells, it's also found in regular cells for their normal functions. Some of these include, as you'll see on your screen, germ cells, which are found in the testes and ovaries, fibroblasts, which are crucial to the structural maintenance of the cell, lymphocytes, which are involved in immune system regulation, stem cells, which could actually be any kind of cell, and epithelial cells, which line the intestines. So the reason this is important is because we are acknowledging the fact that telomerase has functions in cells that are not cancerous. So what this means is that compounds that can target telomerase in cancerous cells can inherently target telomerase in regular cells. And this is a problem because we do not want to denature telomerase in these regular cells.
Okay, so now we're going to describe what exactly happens to a cell to make it cancerous. So the key point to take away from this is the fact that in order for a cell that makes it past crisis to actually survive past crisis, it has to have an oncogene expressed, which basically is a cancer-coding gene. It's a gene that's expressed when the cell is defective. And it also has to have telomerase activation. Now, as you see on your screen here, this figure shows an oncogenetically transformed cell can still die after senescence if there's no telomerase activation. There must be telomerase activation for this cell to become cancerous because that allows it to be immortal and for its telomeres to be rebuilt. So, based on this, there's also empirical evidence to prove that this is true. 85% of tumor samples tested show increased levels of telomerase. Of course, this is also a potential problem when dealing with false positives. As we know, certain cells like lymphocytes, which are often tested for cancer, do have normal levels of telomerase for their regular functioning. So researchers are working on ways to decipher which levels are actually cancerous and which levels are not cancerous.
So in terms of the potential treatments that can thwart the development of immortal cancer cells, As you'll see on your screen, we have a compound called 2,6-diaminoanthraquinone, whose functional groups are NH2s, and it has oxygens on the side here. And we also have G quadruplex, which is a nucleic acid sequence found in the telomerase, and it's actually found in the active site of telomerase, so it's very important for the catalytic reactions that telomerase is involved in. So the G quadruplex has hydrogen bonds, which are intermolecular forces within the molecule, and then there's this positive metal cation in the middle. So essentially, when the 2,6-diaminoanthraquinone comes in contact with the G quadruplex, it disrupts both the hydrogen bonds that exist within the molecule and the metal cation's attraction to the carbonyl groups, groups that exist in the molecule, and as a result, the G quadruplex starts to deconstruct, and the telomerase in general starts to denature. And this is because the active site is very important, as we mentioned, to the catalytic reactions of telomerase. So if telomerase is denatured, then we can no longer construct telomere sequences, and if this is true, then the cell is no longer immortal. If the cell is no longer immortal, there is no more cancer. So that is why this is a potential treatment for cancer.
Okay, so as I mentioned before, there are two potential problems with these treatments. One potential problem is the fact that telomerase activation can be detected in normal functioning cells, which can lead to false positives. Of course, there are different levels between cancerous cells and normal cells and telomerase activation, so this can help people decipher which ones are cancerous, which ones are not. And also, like we said before, this drug on its own is not specific, does not exclusively denature telomerase in cancer, so it could potentially have toxic effects on regular cells. But of course, with the development of drug delivery systems that can activate specifically for cancer cells, this problem can actually be reversed.
So despite the unintended consequences of using 2,6-diaminoanthraquinone and analogous compounds to denature telomerase in cancer cells, it's still a revolutionary treatment, and it's a lot less invasive and potentially threatening than other treatments that have been used in the past. Just a general takeaway from this presentation, we learned about the structure and function of an enzyme called telomerase. We learned about its synthesis reaction for the formation of telomeres, and as a result of studying its structure and function and the synthesis reaction, we were able to discuss 2,6-diaminoanthraquinone's interaction with the intermolecular forces that exist within the G quadruplex, which is a nucleic acid sequence found in the active site of telomerase. And as a result of all of that, we were able to discuss some negative and positive implications of this potential treatment on cancer. So thank you for listening, and have a great day.